Introduction to Molecular Biology. In this lecture, you will, first, explain some of the major milestones in the history of molecular biology. Second, describe the general features and structures of nucleic acids. And third, analyze the central dogma of molecular biology, including known exceptions. These learning objectives will guide our exploration into the molecular foundations of life. So, what is molecular biology? It is the study of biological processes at the molecular level. This field focuses on key biomolecules, DNA, RNA, and proteins, and investigates their roles in cellular function, growth, and reproduction. By understanding these molecules, we can decode how life operates at its most fundamental level. The roots of molecular biology began in the 1860s. Gregor Mendel proposed the three basic laws of inheritance, introducing the idea of the gene as a unit of heredity. Simultaneously, Friedrich Miescher made a monumental discovery. He isolated a phosphorus-rich substance from white blood cells, later known as DNA. Together, their work laid the intellectual foundation for what would become modern molecular biology. Mendel's work gave rise to three core principles of inheritance. First, the law of segregation. Each parent contributes one allele for every trait, and these segregate during gamete formation. Second, the law of independent assortment. Traits like flower color and seed shape are inherited independently of one another. Third, the law of dominance. Some alleles mask the effects of others. It's important to note, real-world inheritance can be more complex, with phenomena like incomplete dominance and gene linkage. Friedrich Miescher was the first to isolate what we now call DNA. He fractionated white blood cells and discovered a substance he called nuclein. This material was acidic, phosphorus-rich, and located in the nucleus. Later, Richard Altman renamed it nucleic acid. At the time, Miescher didn't know its function, but he had discovered the physical basis of heredity. In 1920, Archibald Garrett studied a genetic disorder called alkaptonuria, where patients' urine turned dark due to an enzyme deficiency. This was one of the first links between genes and enzymes. Later, Beadle and Tatum used bread mold mutants, known as auxotropes, to show that a single gene could determine the production of one enzyme. This became known as the one gene, one enzyme hypothesis, later revised to one gene, one polypeptide. Though an oversimplification, it helped bridge genetics and biochemistry. In 1928, Frederick Griffith conducted an experiment using Streptococcus pneumoniae. He discovered that genetic material from a heat-killed, virulent strain could transform a harmless strain into a deadly one. This phenomenon, which he called transformation, offered strong evidence that a heritable substance could pass between organisms even after the donor cells were dead. Griffith worked with two types of bacteria: S strain, which had a smooth capsule and caused disease, and R strain, which lacked the capsule and was harmless. When live R bacteria were mixed with heat-killed S bacteria, the R cells were transformed into virulent S-type bacteria. This provided the first direct evidence that a chemical substance could carry hereditary information. Importantly, Griffith's transformation was reproduced without mice, using only bacterial cultures. In 1944, Avery, MacLeod, and McCarty analyzed the chemical nature of this transforming principle. They found that it was made of DNA, protein, and polysaccharides. But which molecule was responsible? The answer was just around the corner. To pinpoint the hereditary molecule, Avery and his colleagues purifying each component, polysaccharides and proteins were ruled out. The transforming activity was not destroyed by proteases or RNAs, but it was destroyed by DNAs. This clearly identified DNA, not protein or RNA, as the transforming factor and the molecule responsible for heredity. Further experiments by Avery, MacLeod, and McCarty showed that the transforming principle is not affected by proteolytic enzymes or RNAs. However, when DNA was introduced, the transforming activity was lost. This finding led to a groundbreaking conclusion. DNA is the transforming factor and the true molecule of heredity. In the early 1950s, Rosalind Franklin and Maurice Wilkins used X-ray diffraction to study DNA. Their data suggested a repeating helical structure. In 1953, James Watson and Francis Crick built a model that revealed the double helix structure of DNA. The model explained how genetic information could be stored and replicated. This discovery became the cornerstone of modern molecular biology. DNA and RNA are types of nucleic acids made from repeating units called nucleotides. DNA contains the sugar deoxyribose, while RNA contains ribose. These sugar molecules form a ring structure and are central to the nucleotide's backbone. The difference between ribose and deoxyribose lies in one oxygen atom. Ribose has a hydroxyl group on its 2-carbon, while deoxyribose lacks it. This small structural change affects the stability and structure of RNA and DNA, making DNA more stable for long-term information storage. Both ribose and deoxyribose form 5-membered furanose rings through internal chemical bonding. This ring formation is critical for the overall 3-dimensional structure of nucleic acids and allows base pairing to occur effectively between nucleotides. Nucleotides consist of 3 parts: a nitrogenous base, a sugar, and a phosphate group. The nitrogenous bases are categorized as purines, with 2 rings, including adenine and guanine, and pyrimidines, with 1 ring, including cytosine and thymine. When a base attaches to a sugar via a glycosidic bond, it forms a nucleoside. The pyrimidines found in DNA are cytosine and thymine. In RNA, uracil replaces thymine. These single-ring molecules pair specifically with purines in the double helix: cytosine with guanine and thymine, or uracil, with adenine. The purines in nucleic acids are adenine and guanine. These larger, 2-ring bases pair with pyrimidines, adenine pairs with thymine in DNA, and with uracil in RNA. Guanine pairs with cytosine. These base pairs are essential for maintaining the double helix structure and for accurate base pairing during replication and transcription. A major difference between RNA and DNA is that RNA uses uracil in place of thymine. Uracil pairs with adenine during transcription. This substitution, along with the use of ribose sugar, makes RNA chemically different and more versatile in cellular functions like protein synthesis and regulation. A nucleoside consists of a nitrogenous base bonded to a sugar molecule, either ribose or deoxyribose. The base is connected to the 1-carbon of the sugar via an N-glycosidic bond. This structure serves as the core unit for building full nucleotides, which are formed by adding phosphate groups. A nucleotide is formed when 1 or more phosphate groups are added to a nucleoside. The phosphate can attach to the 5-hydroxyl group or the 3-hydroxyl group of the sugar. These nucleotides are the fundamental building blocks of both DNA and RNA. They also play critical roles in energy transfer, signaling, and metabolism. Nucleotides are not limited to DNA and RNA. Important examples include ATP and ADP, which carry energy in cells. ATP, or adenosine triphosphate, has 3 phosphate groups and is the energy currency of the cell. Another example is cyclic AMP, or CAM, which functions as a second messenger in various signaling pathways. These molecules show how versatile nucleotides truly are. DNA, or deoxyribonucleic acid, is a linear polymer of deoxyribonucleotides. Each DNA strand has a directionality defined by its 5' and 3'. The nucleotides are connected by phosphodiester bonds, which link the 5-phosphate of one nucleotide to the 3-hydroxyl of the next. This backbone gives DNA its structural stability and integrity. In this linear chain, the order of nucleotides encodes genetic information. Frederick Sanger proposed the sequence hypothesis. Just as polypeptides are linear chains of amino acids, DNA is a linear chain of nucleotides. The hypothesis suggested that nucleotide sequences determine amino acid sequences, laying the groundwork for understanding the genetic code. In the 1940s, Erwin Chargaff analyzed DNA-based composition from different species. He found that adenine always equals thymine and guanine always equals cytosine in double-stranded DNA. These relationships are known as Chargaff's rules. G equals and A equals T. This discovery was key in helping Watson and Crick build the correct DNA model. Rosalind Franklin and Maurice Wilkins generated X-ray diffraction patterns of DNA. These images revealed a repeating helical structure and helped distinguish between two DNA forms: a form found in drier environments, and B form, the more common biological form, observed at higher humidity. These patterns gave crucial clues to DNA's three-dimensional structure. Watson and Crick used Franklin's data to construct the first accurate model of DNA. They proposed that the DNA molecule forms a double helix, held together by hydrogen bonds between base pairs. Adenine pairs with thymine using 2 hydrogen bonds, and guanine pairs with cytosine using 3 hydrogen bonds. This pairing ensures faithful copying of genetic information during replication. Several key features define the Watson-Crick model. 2 DNA strands twist around each other in a right-handed helix. The sugar phosphate backbone is on the outside, while the base pairs stack inside, forming the rungs of the ladder. The diameter of the helix is 2.0 nanometers, and it repeats every 10 base pairs. Another important feature is that DNA strands are antiparallel. One runs 5 to 3, while the other runs 3 to 5. This orientation is essential for processes like replication and transcription. Additionally, the helix has alternating major and minor grooves, which are critical for protein binding and regulatory mechanisms. The Watson-Crick model also explained how DNA replicates. Each strand of the helix acts as a template for the formation of a complementary strand. This semi-conservative model of replication ensures that genetic information is faithfully passed on to the next generation. It was a revolutionary concept that connected structure with function at the molecular level. This slide references a video that summarizes the structure and function of DNA. As you watch the video, focus on the key structural components, such as the double helix, base pairing rules, and the importance of antiparallel strands. These visual cues will reinforce what we've discussed so far and prepare you for the central dogma of molecular biology. The central dogma describes how genetic information flows within a cell. It begins with DNA replication, where DNA is copied to make more DNA. Next, transcription converts DNA into RNA. Finally, translation decodes RNA into a protein. This unidirectional flow, DNA to RNA to protein, is the foundational principle of molecular biology. To break it down, DNA is transcribed into messenger RNA by enzymes like RNA polymerase. This mRNA then exits the nucleus and is translated by ribosomes into a polypeptide, or protein. Each three-letter codon in the mRNA corresponds to a specific amino acid. This process explains how genetic instructions are expressed as functional molecules. Although the central dogma holds true for most organisms, there are important exceptions. Some viruses, like retroviruses, use an enzyme called reverse transcriptase to convert RNA into DNA. Retrotransposons, mobile genetic elements, also follow this reverse pathway. These exceptions not only deepen our understanding of biology, but have also become powerful tools in molecular biology research. By 1965, scientists like Marshall Nirenberg had cracked the genetic code. They showed that the sequence of nucleotides in RNA could predict the sequence of amino acids in a protein. The code is nearly universal. From bacteria to humans, most organisms use the same codon system. This universality underscores the shared evolutionary heritage of all life. DNA is not just a static blueprint. It is a dynamic molecule that responds to environmental signals. The field of epigenetics explores how chemical modifications, like methylation, can regulate gene activity without changing the DNA sequence itself. Epigenetic changes can influence development, aging, and even the risk of diseases like cancer. Genomics is the large-scale study of entire genomes, all the DNA in an organism. Thanks to advances in technology, we can now sequence entire genomes quickly and affordably. This has revolutionized medicine, agriculture, and evolutionary biology. It also enables personalized medicine, where treatments can be tailored to an individual's genetic profile. CRISPR-Cas9 is a revolutionary gene editing tool derived from a bacterial immune system. It allows scientists to cut and edit specific DNA sequences with precision. CRISPR has enormous potential to correct genetic disorders, improve crops, and study disease mechanisms. However, it also raises ethical questions, especially when it comes to editing the human germline. With gene editing comes responsibility. Recombinant DNA technology opens the door to treating genetic diseases like cystic fibrosis, Huntington's disease, and Tay-Sachs. But it also raises concerns about genetic manipulation of embryos, privacy, and health insurance discrimination. As the science advances, so too must our discussions about what is ethical, equitable, and acceptable.